Hello everyone, this is Professor Taupade Nitin. From College of Technology, Nasik. Today we're going to discussing about the processing technology of beverage practical. Uh, it is regarding to the detection of the E. coli beverage in the beverage. E. coli is nothing but the Escherichia coli. It is a type of bacteria that is normally live in our intestine, and it is also found in the gut of some animals. Mostly E. coli are harmless and even keep your digestive system healthy, but some strain may be causes severe disorder, such as diarrhea, or we can say vomiting, and also some acute kidney failure, or we can say urinary tract disorders. So there is one especially one bad strain, that is 0157 H7, which is very dangerous and which cause abdominal cramps and vomiting, or also bloody diarrhea. So that is why we have to detect this E. coli in the beverage sample. So, let's move with the relevant information. So there are two bacteria that are found in a beverage, or we can say the water sample, that is coliform bacteria and E. coli bacteria. So coliform bacteria are rod shaped and gram negative, and which do not form any spore, which can be fermented lactose with the production of acid and the gas when incubated at the 37 degrees centigrade. Similarly, E. coli are gram-negative, facultative anaerobic, which are also rod in shape, coliform bacteria, and produce indole from the tryptophan at 44 degrees centigrade within the. Uh, so this is about the E. coli. So these are the cultural media that are required for the detection of the E. coli into the sample, that is lactose TTC agar with the sodium heptadicyl sulfate, and the tryptophan broth, and the tryptophan soya agar. So this will require to develop our media, and or we can say the strain. So let's move with the principle. uh, Lactose TTC agar with the Tergitol 7 is actually recommended as a selective and differential medium for the recovery of injured coliform organism from the water by the membrane filtration technique. It is also inhibit the gram positive bacteria, and the lactose fermentation by the coliform result in the development of yellow colonies, and non-lactose fermenting organism produce blue colonies. So this is what the basic principle behind it, which shows the which indicate when we treated with this, when we grow the culture with this agar, it's developed the yellow color in case of the coliform, and if suppose there is E. coli bacteria, then the blue colonies will be produced. So this is a procedure. So first of all, we have to disinfect the surface of the bottle, pouches, and the cups containing sample with the 70% ethanol. So we have to um, disinfect whatever we going to use the utensils for the procedure with the 70% ethanol to inhibit the unwanted growth of the microorganism. Thoroughly mix the sample by the vigorous shaking to achieve the uniform distribution. Then second one, filter the sample through a sterile membrane which has the pore size for 0.4 micrometer, and place the filter on the lactose TTC agar plate. So after that we have to incubate at 36 degrees centigrade for the 21 hour. Then after that we have to examine the membrane and count the characteristic colony of lactose positive bacteria showing yellow color development in the medium under the membrane. If suppose yellow color is developed, then it is indication or the results that coliform bacteria are present. In case of no typical growth after 21 hour, then we have to further extend the time period, which is up to 44 hours at 36 degrees centigrade. Again, we have to examine the membrane for the lactose positive colonies, irrespective of the site, showing yellow color development under the membrane. Then further subculture the presumptive colonies on the non-selective agar, that is we using like tryptophan soya agar, and again we have to incubate at the 36 degrees centigrade for the 21 hours to carry out the oxidize test. Also inoculate the presumptive colonies in the tryptophan broth tube at the 44 degree centigrade for 21 hour. And again we have to examine for the indole production by the adding Kovac reagent for the development of cherry red color at the surface of the broth, confirm the production of the indole. Then 8, consider all colonies giving negative oxidized reaction as coliform. Then we have to consider all colonies giving negative oxidized reaction and positive indole reaction as the E. coli. If suppose there is blue color is developing and which shows the indole reaction, positive indole reaction, so it is indicate that E. coli presence. So we can see this lactose positive microorganism forms yellow orange colonies with the yellow surrounding and have a yellow dot under the membrane filter. Tergitol 7 inhibit the growth of positive colonies and minimize the swarfing of the protease. Further differentiation of the E. coli and the coliform with the oxidase and indole test as required. So in the slides we can see the red dot with the presence of the E. coli, and the second number Petri plate shows the E. coli and the coliform from the water waste, that is yellow color dots which indicate the presence of the coliform bacteria, and those red or bluish color which shows the E. coli content. So these are the results, that is presence and absence of E. coli in the coliform given after confirmation of the presumptive colonies in the sample examined. So test for the E. coli and coliform, that is which is equal to present or present, and which is divided by the absent per xml of the sample. So whatever volume you are taking, we have to divide it with the present one. So this is the observation table. We have to, first of all we have to take two Petri plates, that is, then we have to count out the colonies present in the plate number 1 and the colony present in the plate number 2. So whatever the readings will come there, so we have to put in the final result, or we can say the observation table, that is total plate count of the given sample, maybe the XYZ, whatever we have get after the incubation period. So that will be the our E. coli and the coliform count of the sample. Thank you so much.